Good morning, afternoon, evening to everyone. Um, I'll start. Let's see if this works. Good. I'll go over just some definitions of forensic entomology, the utility of arthropods in legal investigations, the field where arthropod science and the judicial system interact, and certain forensic entomology investigations are utilized by law enforcement agencies. I'll go into it a little while. The study of insects and their use in legal investigations is forensic entomology. It's the link between ecology, science, and the law. It's used to determine postmortem interval, where the elapsed time of death exceeds 72 hours. Sometimes it's commonly termed a minimum time of colonization and the period of insect activity. And I'll show a little later the differences with these definitions. The three major disciplines, and I work in three of them, is urban entomology, stored products entomology, and medical legal entomology, which is the one involved with homicide investigations. Um, so I covered a little bit of the urban entomology here, stored products, possibly a bit, and basically it's going to be medical legal. Also, there's a lot of definitions of it, let's say, medical criminal, and before it was forensic medical entomology. It's a little more specific now. The history of it with the homicide investigations was in China, 907 and 960. The most infamous case in the 13th century China was reported by Sung Tzu, the judicial administrator. What had happened there, there was a death in a field. The person examined the sides of all the people and noticed that flies were attracted to one in particular because blood was on it. It had roots in theory of spontaneous generation. It was more used in Europe mid to late 19th century. Orfila noted patterns of insects that visited decaying corpses. Bergeret in 1855 first used insects to determine time of death in a mummified infant. And Menin started to develop his theory on successional waves, published in La Faune des Cadavres in 1894. And it's been used for many years in the US and Canada. So, as I mentioned earlier, the postmortem interval or time of death is what everyone's always looking for. The minimum time of colonization and period of insect activity um, is time after death and insect colonization, and they won't occur all at the same time. So you have to realize that there can be a death. Uh, there might not be insects attracted immediately. There can be homicide and the person cut up and maybe frozen or refrigerated and then placed out. So your time of death isn't when the insects get there. It actually occurred hours or days before. The insects found on bodies can help with the geographic location. At the time of death, there are different sites of trauma on the remains, and those may very well attract females that oviposit or larvaposit, as is the latter case with the flesh flies. They don't deposit eggs, they deposit first instar larvae on wounds, let's say. And reviewing the larvae, you can also see about the toxicology. Since they've been feeding on the tissues will have toxins if poisons were introduced into that person. So now the larvae can be tested if you can do that in your lab. So here's just a visual on the decomposition timeline, starting at the time of death, fresh bloat, active decay. You have a lot of biomass loss because of that, advanced decay. You have putrid and then dry remains. And at these different times of decomposition, there are different insects attracted. Um, to the corpse because that's what attracts them. Fresh tissue doesn't attract dermestid beetles, but dried tissue does, let's say. So in studies, forensic entomologists have used small piglets, and also larger pigs too, uh, to test and study the insects that are attracted to corpses over what period of time. And as you could see through the past slide and this one, different stages of decomposition takes a certain amount of time, and that's dependent on relative humidity, but also more importantly, temperature. Because warmer temperatures will speed up an insect's metabolism, cooler temperatures slow it down. Why use forensic entomology? A human cadaver can lose 60% of its mass to maggots in just as little as 7 days. You can see this pig here is 3 days post-mortem, and it was in a warmer time of the season. The flies were very active, oviposited, and their first generation has progressed through 1, 2, and 3 larval instars. So the importance of an entomologist at these scenes, of course, would be someone who understands the insects that can be found, um, proper identification of species, and also access to journal articles and a search database. And also in museums, we'd be able to access the museum collections to be able to compare specimens to the unknowns that are known from your collection site at the homicide investigation. And that would help also in confirmation of identifications you've made. The methods for determining the postmortem interval is maggot age and development, and it's based, of course, on these immature stages of the flies. Many of the flies are called, immature flies are called maggots. Some arrive immediately after death, the females, in order to lay eggs, or larvae, as I stated already, with sarcophagid flies. The black soldier fly, which is the image I have here, are attracted to decaying animal plant decomposition and actually are used in recycling organic waste when they're not in homicide investigations, let's say. And as I said earlier, many of the eggs are and larvae will be laid and deposited in wounds or natural orifices rather than just on the skin, let's say. The patterns of succession are the colonization and extinction events. So oviposition, deposit of eggs, larviposition, depositing larvae. Colonization is time of egg deposition or even first instar larval deposition. Extinction is when that insect species is no longer present on the body in its life stages. Maggot age and development is one, here's some of the methods for determining PMI. Mostly there are three families of flies: calliphora blowflies, sarcophagid flesh flies, and mussid house flies. But I'll go into a little later that there are other flies that are attracted to decomposing corpses as well. It's not just these typical ones. So here's oviposition. You have hatching, growth, and feeding. You have, here's some plump, large larvae. These would be third instars. And then you have a prepupal stage. This is when the later prepupal, well, it's the late stage maggot going toward the pupal stage has this migration behavior. So they leave the corpses and will go many feet or yards from the corpse in order to find a great pupation site. And speaking of pupation, most people learn that in entomology. You have larval stages, pupal stages, and the adult stage. But in these three particular fly families and others, it's actually called pupariation because that third larval instar uh, going toward the pupal stage actually uses a third larval instar skin. That's called the puparium. So the pupa develops within this. And you'll find, as you can see, the dark brownish objects are the more mature puparium, and the pupa is actually developing inside that. Here you have the emergence eclosion from that pu puparium, and the adult fly developed inside in the pupal form, and now it's time to get out and do what it does best, is feed a little bit, but mate and lay more eggs or deposit uh, larvae. This is a sarcophagia right here. Temperature and development in insects. You can see the duration of development over time. It speeds up when the temperature is higher by that top curved on the graph. And we actually do this in degree days, and I explain a little later about that, um, in order to figure out how long something has occurred. You're accumulating degree hours, and that's translated into days, let's say. So the degree days, you take the minimum and maximum temperatures for the day and divide by 2 to get an average or mean temperature. If the result's greater than the threshold temperature or the base temperature for development of these particular species, and you have to know, of course, which ones you're working with, you subtract the threshold temperature from the average to get the accumulated degree days for the 24-hour period. If the average temperature did not exceed the threshold temperature, then no degree days were accumulated for that time period. Let's say if insects slow down enough because something happened, it was cooled, like after they were in um, on the corpse, maybe someone took the corpse and then refrigerated it, and that would throw your time period off because the temperatures did not allow progression in the life history of that fly or flies that are larvae are now on the body. So here's just showing a generalized life cycle for the caliphorid fly bottom and the Hermetia, which is the other fly. Okay, so now maggot age and development, analysis of insect stage of development plus temperature records, and that will convert to accumulated degree days. And uh, let's see, so you get the analysis of insect stage plus temperature records plus species, and you get the date of eggs or larvae when they've been deposited, which is the minimum time of colonization and the period of insect activity. So here's a bunch of different images. I'll go into some of the larval morphology too later. Here's a maggot showing the um, top section here, the mandibles, their modified structure for chewing and scraping, and the rear section is for the breathing in the maggot. And you can see the fly is emerging from the puparium here. The tilinum, the front section of the head, actually pops the cover out, and that's why it's expanded in that specimen. This shows the diagrams, different families on the right with sepsids, sphericerids, piophilids, and phorids, in addition to the other ones I spoke of earlier, and the some morphology of these different maggots are also shown. And the breathing system, um, when the first instar, you have these plate was actually one slit, second instar two, third instar three. And here's variations of these different spiracles with the slits of different families, different species of various flies. Very useful for determining the species. There's a new key that came out. It was an interactive key for Californias. This came out of in Brazil. It's online. You can actually use it interactively. Adult characteristics, you can see the fly head, eyes, compound eyes, antennae, and various setae, and palps, which are important for determining the species you have. The bristle position of these two Lucilius species are very distinct, with two here and three here, and that helps for identification. And you can see from all these uh, uh, setae on the body, they're each are identifiable with specific names for where they are on the body, and that's used for determining the species too. Here's thorax, uh, underneath the ventral view of the abdomen on the right, legs. Wing venation is very distinctive too, and that helps for determining the species. And here's as I mentioned earlier, you can see a fly that emerged and is darker and it's more sclerotized, but the new one that just emerged, you can see its head is pale, body's pale, and the tillum sticks out forward because that was used to pop the cover off of that puparium. And here's a methods to determine postmortem interval with patterns of succession, the oviposition or larva position, colonization, and extinction, which I went over earlier, successional waves of species present at discovery, previously present, and that have not yet arrived, um, because these are supposed to occur. And that helps also in determining time of infestation. And in addition to the flies, of course, there are different insects too. So you have different beetles are attracted, different wasps are attracted. The tiniid moths are later on when the body is dry. Some of the beetles then are also attracted later on when the body's or muscle and mass is drier. And like the staphylinid beetle that's here, that's actually attracted as a predator and will feed on some of the insects that are there. In addition to that ant, that's also predatory and will pick out some larvae from the corpse and can throw off the timing because you have lots of ants there. They're taking all these insects away from the corpse and they're not going to be decomposing that corpse any longer. You also have mites are attracted. And they might be on some of the insects, riding on there like a bus. And then they're brought to the area, to the corpse that way. There's a whole review of which flies and which waves of infestations exposed corpses um, and had, let's say, the witch flies, the state of the corpse, and what age it would be at this point, and different creatures come in different waves. And as I mentioned earlier, there are different insects, arachnids, and myriapods associated with the corpse that aren't necessarily um, those like the typical flies that are attracted, but they're in the environment and they come out and investigate this new meal source, but not typically on dead bodies, let's say. The one little booklet on forensically important insects was done years ago, and here's a review of some of the information, the pictures of various creatures, insects that were attracted to various corpses. And these can also be these are the other arthropods that are not the typical ones. Um, and depends if the person's in the water. Let's say you could have various aquatic insects now attracted to that corpse. You have spiders are attracted to a corpse because they're preying on the insects that are there, and some even build webs on the corpse to pick up things. And here are various true bugs on the right, and wasps, bees, mayflies, and flies, cockroaches, ants on the left. Different fly species here, some of which I've gone over already in these reviews. And here's some caliphorid flies, sarcophagid flies, metallic ones are caliphorids, the dotted ones here are the sarcophagids. And you could see there's some larvae, first instar larvae, that were dropped from this female fly because she was stuck. And what they often do is get rid of their eggs or next generation load if they're stuck and they're going to be dying. And the reason they were on this glue board is because there were mice stuck on here and they were attracted to the decomposing odors. Factors that influence forensic entomology, a geographic region, season, habitat, the maggot mass, any drugs and toxins I went over earlier, certain clothing, if it's totally wrapped on the corpse, maybe the insects really can't get to it, and condition of remains, and there's various ones listed there. Now in sample collections, there's many different materials that are used, jars, vials, alcohol, different kinds of alcohol, live containers if you're going to rear them to get adults. Now this might be also on apps, and this is just a paper for noting information of various variables when collecting insects. Here are some ways of collecting and pinning them, labeling them, or keeping them in alcohol, depending on the specimens you have. Some tiny things you might need one of these aspirators to suck them up. Sometimes if you're at a corpse and it really is a bad smell, yes, you have a battery-operated aspirator. Sorry I'm moving quickly to get through the presentation, but here's ways of rearing them too. And this way is if you're getting material, let's say from a leaf litter, you use a little heat source, a Berlese Tolgren funnel, that makes the creatures leave that source, fall down the funnel into the alcohol in the jar below. And also egg collection is important. 96%, 75% ethanol is typical. In some DNA work, you might want to use 96%. That preserves them much better. And here are some maggots and some first instar larvae that have hatched. We went over a part of this before, but just because you get large larvae doesn't mean they're the oldest ones who've been there the longest amount of time. It's also important to keep different areas of collection you want to keep separate, not just put them all into one jar. Here's an outdoor case just showing you. This was done at the Body Farm in Knoxville, I believe it is. And this was just to show over a time period how insects infested the body and decomposed the body. And then an indoor case was up in Manhattan in an apartment just to show you some of the flies and where the maggots were found in some of the orifices, like in the ear. There's where some were found. Under the hair, behind the ear, you can see some maggots there. These were a little younger maggots, so this was not at the third instar and they were going to leave. This was up until that point. And I also tested with some um, students back in 2008. We used a citrus to fix the tissue in the maggots before putting in ethanol preservation. Because normally if you were to take maggots, put them in 75% ethanol, you might get some decomposition and their maggots aren't plump. But using this system, actually, instead of using boiling water if you can't use it, this citrus material worked pretty well on maggots. And we tested them out from different lemon, lime juices, to citric acid, um, vitamin C, and it worked pretty well on vitamin C. It was useful and it worked pretty well. Um, you can read this, it explains it. Uh, the vitamin C maggots, though, weren't tasty compared to the ones with the fruit juice, the citrus juices. I work in edible insects, so this was a little bit part of that, not just forensic work on corpses. And the maggots were not from corpses for this test. They were just reared from a laboratory colony. And collecting them, um, we keep them upright, package them, breathable material. This is if we're using live ones, of course. And the rearing, um, we can use different materials, beef liver, pork liver, and breathable containers, of course. Here I had a little thermometer humidity indicator to see how they were doing. And here's some of the larvae and puparia. You can see the different stages in the puparia life because they're pale to dark. So they're not all produced at the same time. And here's the puparium. You don't have to preserve them in ethanol. You keep them alive because you want an adult from there. That's much easier to determine the species than a larval form. And here's beetle collection, various beetles associated. This was a book next to a corpse, and the beetle larvae left bored in to pupate. So they have pupation chambers inside. Here they are. And even rodent traps. These had domestic beetles, and the larvae had skeletonized the dead mice in there. This was from an apartment. And here's the adult beetles and shed skins, black carpet beetles. Those are the ones in homes, very common. And what else can the insects tell you? If a body's been moved between locations, habitats, environments, if it's been disturbed, which I went over a little bit earlier, so it might throw timing off. Wounds is where they like to go, and some of the different insect feedings, like the ant that you see here, or mimicked, um, what else was going on. Also, the chemical burns were actually from the ants feeding. It wasn't burns at all. So here's just a little review of that. The fire ants were feeding on wounds on the skin. And the insect parts can link a suspect to a victim location, DNA extraction from the gut contents. And I went over earlier about this, the drugs and toxins, analyzing maggots, pupae, pupal cases or puparial cases. The insects bioaccumulate materials, so they're a good collection site. And the toxins and drugs can speed up or slow down insect development too. And this was poaching was not just homicide. This is another use of the insects. And senior child and um, neglect cases, abuse collectives. This one was recently reported in October 20th about a woman who had been in a, well, here, let's see, she'd been in a reclining chair for about 2 months. Her son was supposed to take care of her and didn't take care of her, of course, even though he said he did. And her chair was covered with maggots, and she had wounds on her body from them, and also just from sitting in her feces and in diapers there for that amount of time. And I know there's a talk about um, dental, oral dental, uh, anyway, uh, later on, but here there's some cases of oral and nasal myiasis, and there's a few cases that I've looked at where the maggots, because of something, either halitosis, wounds in the body, in the mouth, attracted the adult flies to deposit larvae or deposit eggs in there, depending on the fly species. And these are other insects that are associated, although I might not have time to go through them all, but just to show you that other things on people's bodies might be there when the person was alive, such as different lice species feeding on the person, crab lice, body and head lice, fleas, which might be because of pets that are there, not not necessarily human flea, which that can be also. Flea bite dermatitis, we knew this was flea bite dermatitis just because the person brought the fleas in. Otherwise, it's very difficult to determine by skin lesions what actually caused it. Different cockroaches you might find on a body. These are brown banded, these are American, these are Oriental, and these are uh, sh the molted process of an Oriental cockroach male and female. And I worked with one crime scene artifacts with cockroaches. The people, the investigation thought that because of the spray of blood in areas, um, it was from gunshot wounds, when in fact, by rearing the American roaches that were there and other ones that I got from colonies on blood, we realized that the position of the feces actually mimicked the spray of what was thought to be blood from wounds, when actually it was just cockroaches feeding on the body, getting blood, and then defecating on the walls. This wasp here tells you that it's a parasitoid of American cockroach, 8 cases, and that you must have American cockroaches there in order to have this wasp present. Sometimes there are also Oriental cockroaches. And here's a parasitoid wasp depositing eggs. It's in the cockroach egg case. These are bethelid wasps that are ant-like and misidentified as ants, and the wounds on the person were misidentified as bedbug bites because these wasps were stinging the person. So these different skin lesions here were actually the stings and not bedbug bites. And pigeon flies, some of these will come in and bite people too, because they're on the pigeon nests that are out on the AC units on the windows. And here's a close-up of one of these. And on deer, you might have these feeding on people if they're hunters and they hunted deer. These insects could be on the person. And bird mites, also nests in the windows, AC units, and the pigeons are there. And these bird mites were on the person. There were rodent mites. These were collected. That's how we knew what they really were here. And tick species. You might find different reactions from tick feeding on a person and Lyme disease association. And the overall conclusions, forensic entomology has been around for centuries. Maggot age and development are more reliable methods for PMI estimation, and many limitations to methods, but many strengths. Determining the minimum time of colonization. You have to incorporate error rates into estimates and recognize limitations and factors of influence. And research in improving insect development, successional data patterns, and incorporating ecology of the various species into determining the PMI too. Um, let's see. So here, just to review, the postmortem interval, the minimum time of colonization isn't the post-mortem interval because you have also the period of insect activity. The have to be figured out in order to give you the proper post-mortem interval. Just because you find them doesn't mean that's when the person really died. And since it's late, I'll skip the section on bed bugs and rearing and some other items associated, except to tell you that here I noticed when putting them under a microscope with white light, the adult bed bugs on the top looked like adult bed bugs. The ultraviolet light, the resolin area on the ventral section of the bed bug uh, became bluish. And that's the part that expands when the insect is feeding. So that we figured that must be resolin reacting to UV light. The interesting thing, though, was infrared light that actually cleared the specimen and you could look right through it. And it looks like it was made of clear glass. You can watch the alimentary tract uh, moving as even blood was being sucked up by this insect on a live specimen. So that's just something there. And we worked on the genome assembly in the bed bugs. And also, just to tell you that bed bugs, since they feed on the person, um, they can be used in forensic for blood samples because bed bugs feeding on a person there, let's say the corpse isn't there anymore, bed bugs are there and they're plump. collect them and review the and run the blood sample through to give you a profile of what's in its gut. Because it can work up to at least 2 days and maybe even longer to give you a viable um, result from the blood that's inside the gut of that bed bug. And there were some papers here on it. And just with the feeding, and just actually to say one other thing, if you come across a site and it's infested with bed bugs, don't sit down on any place. Even if you don't see them, the heat from your body will attract them. Your carbon dioxide that you exhale will attract them, and then you'll be another harborage site of bed bugs, which usually you don't want to happen. Thank you. And just some reaction bed bug feeding I've done. I've done it for 40 some years, so I fed about 500,000 bed bugs over that period of time. And luckily, I don't react badly like other people do to their bed bug bites. So with that, I'll stop sharing and maybe be able to take questions.